Hi, everyone. I want to thank you for inviting me. I am the chairman of a firm called Feinstein Kean Healthcare, which is a strategy consultancy that has spent the last 25 years essentially helping the people at the bleeding edge of a variety of transformations in biomedicine. And I'm particularly pleased to be here today, one, because I think the discipline of getting through a presentation in 5 minutes is something that should spread everywhere. So it's kind of an experiment for me. But also because I think there's great deal of synergy potential between the organization that I had the privilege to found and DELSA. So the things that I think about in this new dawn in biomedical research are 3 things that occupy the attention of the people I spend all my time with, who are both in the biological sciences, the healthcare arena, as well as in the tech world. One is this shift to personalized medicine via the genomics and other omics revolution, which 10 years ago was something people told me I was crazy to be focused on, and now is basically everywhere and transforming medicine as well as science. The shift to collaborative models as we move away from the traditional 300-year-old one man, one microscope model. And the third, which I haven't heard this morning mentioned, is the demographic trends and the public health issues, whether that's cancer and other chronic diseases such as diabetes, that are making the costs of central importance in healthcare and essentially forcing productivity. So I think biomedicine is being brought to the table, kicking and screaming to the digital era, basically, if nothing else, because of the money. We simply can't sustain what we've been doing. So all those 3 characteristics or trends, I think, intersect at something called the learning healthcare system. So can I just see a show of hands? How many of you are totally aware of and facile with the learning healthcare system? Okay, there are a couple of you. So what's the learning healthcare system? It got started here in the Beltway. Basically, the concept is that all healthcare should be delivered according to an evidence base, not the latest publication you just happen to read in JAMA, not your gut as a physician, but that there needs to be a focus on exactly what is the evidence. And that presumes that there is a circular continuum that goes from the bench to the bedside and back to the bench. That's the gist of it, right? Novel innovations and discoveries get rapidly to the bedside where they are practiced, and every single clinical encounter becomes the grist into a giant big data mill that goes back around to the bench to facilitate and accelerate research. And that has, over the last 5 years, gained enormous currency. Lots and lots and lots of people talking about it now. But as this group well knows, there are in biomedicine countless obstacles to the kind of data liquidity you would need to envision a true rapid learning healthcare system, whether it's privacy, standards, IP, blah, 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 blah. Now, the good news is there's a lot going on. This meeting is itself evidence that there's a lot going on, but partly that's what scares me, that there is so much going on. There are a jillion different consortia. There are a jillion different efforts between this institution and that institution, between this country and that country, between this discipline and that discipline. But there is really no entity that I'm aware of that has taken an overarching view of the technical and cultural standards and norms across the entire enterprise. And that, I think, is what we need to really move biomedicine forward. I think we are all looking at different pieces of the elephant, but I'm not aware of any other organization that thinks about the elephant overall. So the Data Liquidity Coalition was formed as a result of a meeting at the Institute of Medicine last year, a workshop, and it is comprised of all the stakeholders in the cancer community where it got started and beyond, who are committed to a common vision of data liquidity to achieve personalized medicine and the rapid learning healthcare system. We go across truly all constituencies that have been mentioned today. Advocacy, education are among our activities. We want to wake the sleeping giant, which is the patient community that has no clue that their data are not being used productively for research. And we, in particular, are going to work on demonstration projects that actually formally address some of these challenges. So I'll stop there. Thank you so much.
Okay, so I'm going to change tack slightly. I'm just going to talk a little bit about the research that my group does, and the only reason why I'm doing that is not to impress you with the research that my group does, but to show you that there are opportunities in data that are not immediately obvious for most of the omics data sets. So we've heard the elephant analogy a few times, right? So people look at different parts of the elephant. I do weirder things to elephants than look at their parts. Okay, so the first thing I try to do is I try to take the elephant to new places and see how they behave and what happens. And the second thing is I try to get very many herds of elephants together and then go and look for a particular mold on one of the beasts, right? And so that's what we're going to try and show you. So proteomics is mostly driven by mass spectrometry. And the nice thing about the mass spectrometers these days is that they're incredibly sensitive. They can find out a huge amount of stuff in your data. The sad thing about it is that, so that's the top line there in blue, proteins, peptides, spectra. The way we get results from these spectra is we actually start from known stuff. We start from protein databases about the sequences we know. We generate in silico peptides, which is dull and boring and easy. We generate in silico spectra, which is dull and boring and easy, but should be made a lot more interesting, and we're trying to do that. And then we match the two. So we can only find and identify those spectra for which we already know what they could possibly be. That means that anything that we don't expect will be missed. You know, a priori, you know you're going to miss it. So our results are always limiting. I'll show you how limiting they are. This is data from the repository I built at EBI called Pride. Data is a little bit out of date. We're now closing 400 million spectra. This data is about 8 months old, so you get 100 million spectra in less than a year added. You see all spectra in blue on top, and then you see the ones that are identified. A slight discrepancy. And so what you can do now is you can do two things. You can try and get more out of the 60 million or so identified spectra that we have today. So that's the green bar. But you can also try and do something about this gap between the green and the blue bar. And the really cool thing is that in both cases you can find new stuff. So 2 examples. This is how we try to reuse the green bar. We have done a reanalysis of all recorded phosphorylation sites on proteins. And there are very, very many across all the different experiments. So we've got close to 1 million phosphosites, and we started mapping them on known 3D structures of proteins. And some of them are really beautiful textbook examples, like the one on top. I mean, that looks like a protein docking or recognition site if I ever saw one, right? I mean, if it were a spaceship, you would immediately land there. And there's a phosphosite right in the middle. And you can imagine that that phosphosite does things to the protein and the way it interacts with its fellow proteins. But there are also other ones, like that one at the bottom, that also looks like a weird spaceship, definitely the bad guys. And there in the middle of the protein, in the middle of this bowling ball, this tightly knit bowling ball, there is a phosphosite. So there's 2 options. One is, it's a wrong phosphosite. It's a spurious result. It's a false positive. Second thing is, something happens to this protein. Something weird happens. It may be cleaved off, parts of it. It may refold. It may do other weird and wonderful things, and that phosphosite might be crucial to all of that. So what you can do is you can generate leads. You can say, we've got a million phosphosites. Which of these makes sense? Well, the top one obviously makes sense, but I'm pretty sure there will be extensive literature about that one already. But the bottom one is one that you could potentially pursue. And so you could use this to try and look for things that are very odd. And we all know that in the odd things lie the interesting things. Okay, so that's one way you can try and take existing results. Take the elephant somewhere new. Take the elephant to your local bar and see what happens. The second thing you can do is you can try and get herds of elephants together. And the good thing is that if you get very many of these animals together, you can start looking for things that you cannot see on an individual animal because you have to be so lucky to see something like that. What we do here is we're looking for coding, long non-coding RNAs. Sounds a little bit silly, right? Non-coding RNAs, but if you look at the sequences of the long non-coding RNAs, and this debate has been going on in the literature for a while, some of them seem to have codon usage. Well, do not seem, they actually have codon usage that is influenced by evolutionary pressure. So they have coding style nucleotides. And so we decided to go and look for these things. Now the problem is that in the vertical line, if you make a vertical line, a column, you get a single experiment and you get all the known long non-coding RNAs on the y-axis. Well, you get a sampling of them. There's many more. And in every individual experiment, it's extremely hard to find more than 1 or 2 hits, right? So if you go bottom to top, you will see that there's only 1 or 2 on that column. And if you compare that to what you get if you search complete nonsense, which is a poor man's way of doing false discovery estimations, you will see that you will get equal numbers, right? So in every column you can find 1 or 2, whether you're on the left side or on the right side. So essentially any individual experiment will have a false discovery rate for these guys of close to 100%, if not more. So there's no way you're going to find anything. So what we do is we look at cross experiments, and then these things prop up. And then you see that this particular long non-coding RNA keeps coming back. And that is something you don't see on the decoy side. So if you have enough data across enough animals, you can really start doing these things. So it's just one thing that we do, but I think there's future here for big data to do a lot more than you could ever do with a single data set. Thank you.
I've been blowing the workflow horn for a while, so that's what I'm partially doing, and also in addition to other things. But workflows now are useful, scientific workflows in general, in biomedical sciences, and they are used as toolboxes, doing some research, and in some steps of their pipeline, right? It could be data acquisition, generation, to direct publication of that data, but it could be the analysis of it. It could be from analysis to publication, providing reproducibility-like aspects. So while they're doing this, what do they facilitate? What do they do actually that helps? One of them is basically maybe the most important one for sharing this encapsulated knowledge within a network is really assembly of this complex processing a lot easier. So, and formalizing it as more like understandable blocks of information. And they also provide, while doing that, most of them try to do, and some do it in a great fashion, they provide transparent access to different resources, data resources, computing resources. And while doing that, they incorporate the community tools. And you know, they also provide some support for provenance in turn for reproducibility to a level. But most importantly, for organizations, cyber infrastructure projects, or organizations like DELSA, they actually facilitate this development around the community development model. So they are actually big open science advocates in that sense. You see a lot of workflows in these open science platforms behind the gateways, so they get shared in an open fashion within a community. So of course, while doing that, they foster the collaborations, right? And while doing that, again, they provide some flexibility and synergy between people, and they optimize resources at hand, both in computing fashion, but also people resources. And they increase the reuse of scientific products, and they also provide some standards compliance. So it makes it a lot easier to go from data generation to publication and insert some standards if you have some standardized pipelines for it. But of course, they are not Microsoft Word. You can't just open a workflow system and just start typing, right? You need to know a little bit about what you wanted to do, and it's a little bit of a roadblock for usage of workflows for scientists, because you need expertise to use which tools, both in bioinformatics or life sciences in this domain, but also in the big data domain, in computing domain. And they also require computation models to schedule and optimize these. So it's a roadblock from the computing perspective maybe, but also it doesn't exist a lot of the times when you bring all these expertise that require different parts of the workflow to execute differently. Doing okay, I think, right? So BioKepler is an example of a workflow system. Kepler actually is an example of workflow system, and BioKepler is a module on top of that. So we used Kepler pretty creatively in different systems, but in life sciences, one of the examples is the camera project. In camera, we have actually 30 workflows that are given to the community to build as workflow building blocks for larger studies. So it's not very often that you run one workflow and you come up with results. You actually run a study using workflows, and you go through these in an experimental fashion, really methodically, and you come up with results. You link the results, and in the end, you have a discovery, the hope is. So on these platforms like Camera, which I sort of put as cyber infrastructure platforms, workflows, not just Kepler, any other workflow system can run in the background and be served to the community. In Camera, there are 30 of them, and there's 1,500 submissions of these workflows a month. The number is growing. So, and in turn, they actually run millions and millions of jobs, which actually makes us consume any resource we are given. But there is this bio and compute play that goes into this, and there are best practices that are done by community. How do you bring these together? How do you actually analyze a biological code and see how it runs, what efficient ways it could run using things like platforms like Hadoop or other technologies, and bring them together. So what we are doing is we are building blocks for executing codes and making the programmability of these codes actually a lot easier by the scientists. And again, Kepler is an example, and we had various use cases a study. Wei Zhang, actually, who built various pipelines, is my co-PI on BioKepler. So what we are going to do is then convert them into BioKepler workflows so that we would actually provide them as services, maybe through DELSA to the community, and foster maybe a culture of sharing this knowledge through workflows as building blocks within DELSA. Thank you.
Okay, so heard a lot the last day and a half about sort of the big data problem, right? So what does one think about big data? Thinks large genomic sequences, large data coming off Hadron Collider. But I think there's another aspect of big data that we can't forget, and that's that there's a long tail of data. As the amount of data that's getting collected goes up, the ability to collect that data also goes up in terms of the cost of the technologies. People have now easier tools to build data repositories, databases, and so there's a critical need to be able to sort of find and utilize these data. And it's a little washed out, but there's sort of this, the iceberg metaphor where there's, you see a little bit of the top of the iceberg. That's, what people know about data resources that are available. But the majority of this iceberg is actually underwater. There's a lot of dark data out there. And so what we've been involved in the last 4 or 5 years is to basically build a platform, a data platform for communities to be able to, find these resources, utilize these resources. And starting sort of at the lowest level, that entails simple things like building a catalog, community-based catalogs, as was mentioned regarding 3DVC yesterday or today, in the NITRIC talk. These community resource catalogs where members can go in and find resources that they might not be aware of. As you heard with some talks earlier today, it's really difficult for people to find, what they need. And that entails, some simple cataloging technologies. But then once you have this catalog, to actually, pull in data, to find this data, we have to develop other sets of tools to basically enable communities within themselves to enable search through semantics and other community-specific ways, pushing this data up so that they actually have access to data in the databases themselves. Another issue in that Google and other search engines, common search engines, don't allow researchers to find data that are specifically needed for their research. Google does not do a very good job of indexing content in scientific databases. So how are researchers, researchers can't go to 6 different databases, 7 different databases, learn all the different interfaces, do all the searches together, and then try to collate all that information. So how can you make that easier for them? So what we've done is provided technology that allows our system basically to go in, pull this data out of the databases, start collating that information, add some semantically enhanced annotations so that if you're coming in from a certain domain, that you can actually find relevant data based on topics of interest. Once you have this data then in a common form, you can then start applying other processes. So we heard about nano-publications earlier. This is all built on top of semantic web technologies, RDF, linked open data. So once you have this data in a format, you can then apply these other transformations on top of that data to start publishing it in new and novel ways. And that's very important with a lot of these communities because the people who have built a lot of these resources don't have the funds, don't have the technical expertise to actually do this type of work. So what you're trying to do, and what we're trying to do, is we're trying to enable communities to basically lift their data up through this process so that it's not, a burden on, a small research lab that might have a very important database or set of data, but get the community in there with a set of tools, with a platform that they can go in and start helping and making this data more available and more useful. What I'm going to talk about real quickly is that The sort of flagship project for this was the Neuroscience Information Framework. So this was in the context of neuroscience. So this was funded from NIH Blueprint, and so we've done a lot of work in the neuroscience domain, and that includes, you know, work in building the ontologies to help sort of semantically enhance a lot of this data. But we've now taken this platform and are now applying it in other domains, such as in diabetes and kidney disorder. We just had a recently funded award there that we're starting on, you know, to apply some of the same technologies there. Also in the aging domain and with animal models. So, you know, we're trying to provide sort of a set of services, a basic platform where communities can go in and enhance their data, make it more easily usable, more easily findable by the research community itself. Because again, you know, as Delsa points out, it's not one person's responsibility, it's a community effort. And so how can you enable the community to deal with this long tail of data? And that's what we've been working on. So I think
So I want to come, like you have seen all these plots before, but I want to come from a different direction, like how a computational scientist think about a life sciences problem. So there is an exponential of data growth in genomics, proteomics, from next generation sequencing technologies, mass spec, and so on and so forth. But the big problem is actually data storage, data movement, and also how to analyze this big data to come up with the big knowledge, right? So that is a big bottleneck right now. So that's the area where us computational scientists can get into, because, you know, the exponential data growth is surpassing like Moore's law or any storage or anything like that. So it's another thing we need to really look into is the data movement from gigabytes to terabytes, so how we're moving. Till today, it's done taking hard drives from one place to other place, right? But Internet 2 and Globus Online, they're trying to come up with various software tools and various other entities to solve this problem. So and this data movement is a very big problem because these technologies are going global. So it's not one place. So another thing what we can do is we can take compute to data. So that's one way of solving this, right? So you have various computing centers all over, and you can compute where data is. And as you know, it's getting cheaper and cheaper to sequence a genome, but the downstream analysis is getting expensive because these programs were developed when the data was manageable. Like most of the applications were developed in mid-90s, early 2000s. So that's the problem. That's a very big problem. But and also the computing architectures are getting better and better, right? So we have this next generation supercomputers coming up, so which are in petaflops, and we are going towards exaflops. So where life sciences can contribute is, or DELSA per se, is to change the programming modeling for life sciences itself. So we take a problem at the kernel level so that it's more efficient and can run on this big iron machines, right? So that's where we scientists can help to generate large-scale knowledge discovery, right? So using these supercomputers. One other thing I want to emphasize is researchers like Larry Smart don't have to wait days and days for their jobs to run. So we need to have something like a dedicated life sciences machine, right? Something like Big Red 2 or something like that. But we also need to look into various programming models. So we have this parallel application wrapper where we can run like tens of millions of blast runs a day, hundreds of millions of domain searches a day, or we can dock billions of small molecule compounds to do drug discovery in a day. So we do that on hundreds of thousands of cores on the supercomputers that we have in Exceed or in DOE. So it requires quite a bit of change and thinking and training to actually use these big machines to run millions of jobs and searches, and also science gateways and automated workflows play a major role. And I think DELSA should combine all those in one place so that all life sciences researchers can access these big machines and big workflows from one interface. And as you know, there's billions of dollars funded for the science cases, hundreds of millions for supercomputers, and maybe thousands or hundreds of thousands for applications. But also we need to think about algorithms itself and training next generation students. I love to see more graduate students involved in DELSA, right? So training is also a key issue, and we need to address that here. That's all I have.
Marcia, you talked about data liquidity in rapid learning and biomedical research, and you talked about what might happen when the patient community becomes aroused. Whoop. I was wondering if you could expand on that a little bit and describe how, what exactly you think is going to happen.
Is this on?
Yeah,
It's on? Yeah. Well, it's an interesting thing. We work with a lot of patient advocacy groups, and the traditional model, as you know, is that an advocacy group would be dedicated to one particular disease. Usually the founder suffered from it, or her child did, or parent, and they would devote themselves wholeheartedly not only to educating about the disease, but to raising money. And the money would go to the research community. It would be essentially thrown over the wall. And the researchers, if they were lucky, would say, thank you very much, and that would be the end. And the researcher would go on to pursue his or her goals of publishing and getting the Nobel Prize. But basically, the advocacy group was treated as nothing more than a bank account. And that model has been changing dramatically in recent years, where the advocacy groups now feel that they are full partners, that they develop strategic plans in conjunction with those research entities in terms of how they're going to attack the disease, and most particularly, what is the strategic plan to getting to a new set of treatments and cures. The thing that I have yet to see from any one of those forward-thinking advocacy groups is what they're going to do about the fact that all the data are locked up in silos. Moreover, that one of the greatest assets that the advocacy group itself has through its membership is the clinical data, not to mention the biological samples, from their own membership who have the disease. I think when they, and we've tried to do surveys on this subject to get at it, they don't even understand the question. So once we educate them around what the question means, i.e. where are the data all going and why aren't they unlocked, I think there's going to be first a tremendous sense of outrage, and then a demand, which I think will be systematized in the way they give out grants, to say this is simply not acceptable, and there has to be a pathway for data sharing in a rapid, facile way. I think that's going to take at least 5 years, but we want to set forth on that to see it happen.
I heard a really interesting talk at Science Online in January where there was an ethicist from a hospital talking about patients coming in, driving miles and miles and miles, getting their blood drawn for cancer research, and that they had signed all these forms for the IRB, the consent forms, and she thought how sad it was that that one researcher left and closed up their lab, and all that data was locked away and never shared with other people when the patient had gone to considerable effort and sacrificed quite a bit to make that data.
And the patients have no idea that that is occurring.
Right, so that's good. Sanjay, you had a question?
A couple of quick questions, starting with Lennart. I'll just ask the questions, you can answer at your leisure. Lennart, how many proteins are there today, both annotated and unannotated? Marcia, I think it's a basic comment that I think I mentioned this yesterday, that the personalized health record industry failed because the penetration of involvement of patients is pretty weak. Even today, it's only between 2 and 6%, depending on who we talk to. So isn't this somewhat of a pipe dream that the pipeline should probably be the physicians and not the patients? So I need your comments on that. Ilkay, how do you define a job in Rekepalli? And Bhanu, the questions that I'm getting from some of the very large institutions like Broad is things like eventual consistency. Supercomputers may not solve the job. The question may be commoditized parallelization, both on the storage and the compute side. So, thank you.
Bomb successfully dropped. Shall we go in the same order that the questions were asked? Okay, so you asked me how many proteins are there, annotated and unannotated. The answer is that depends on what you mean by the protein and by the annotation. It's a good thing that Bill isn't here because he would make a big fuss about isoforms. I'll give you some numbers though. If you look at mass spectrometry-based proteomics and the human proteome, which is the most studied, full stop, we have about 20,000 odd protein coding genes in the human genome that we know about, and it's probably pretty close to the full tally. Of those, we have seen at least one peptide in the public domain, which is only the tip of the iceberg, for about 80% of these proteins. So we've seen quite a few of these proteins. I mean, the machines are pretty good and they're getting better every time. And actually, my numbers are already 2 years old. So today, this is a diminishing returns curve, right? So maybe now we're at 85% or so. However, the problem is isoforms. One particular gene can have more than one protein type, and there, unfortunately, we are just scraping 32%. We're very bad at isoforms. It's a big challenge in the field of proteomics that currently hasn't been addressed yet, but it should be on the agenda. When you look at protein structures, because I talked about structures, the figures are a bit more bleak. So in the 70s, it took a very long time to do any protein. Then it became fast for a certain family of highly soluble, easily obtainable, and purifiable proteins. And so now we're at, again, at a diminishing return curve, so sigmoid that's actually leveling off in the middle. And there we have the problem that a lot of these proteins are membrane proteins, very hydrophobic, very difficult to crystallize, very difficult to analyze. And so there we're stuck in a very bad situation. The good thing is that we do realize that we've covered more or less all the possible structural elements in a 3D structure of a protein, which means that if you de novo get X-ray diffraction patterns from a protein crystal, you don't need to do any more stable isotope incorporation and all of that necessarily to come to a structure. You can actually use certain building blocks and homology to come to proteins. But I think we're just waiting for a new breakthrough technology there to push it beyond and to go into the membrane proteins. And final thing I'll say is the membrane proteins are the most interesting. They're the way that the cell communicates with the external world, full stop. And that is where it matters. Okay.
Okay, on the personalized health record question, you really asked a bunch of things. Let me answer first at the most superficial level. I think the first generation of patient health records were really dumb. They generally were not connected to the EHR, and anybody who had the time and the patience to type in their entire medical history and try to draw everything in and pull it from shoeboxes and doctors and x-ray clinics, and oh my God, nobody in his right mind would have done that. But that's not really what I was talking about anyway, I think. There's two questions. One, is the patient data being entered somewhere? And it's got to be tied to the EHR, and I think it's got to be tied to the notion of patient-reported outcomes. So rather than keyboarding my entire medical history, I'd be quite willing to keyboard, did the asthma medication that I was prescribed last week work, or am I still gasping? It would take me about 30 seconds to do that at my provider's portal, and I'd be more than happy to do it because in general, no, they're not, and that's why I'm gasping. The other question that patients in particular need to be thinking about is whether their data are being facilely accessed and distributed in a liquid way, and that's really why I want to mobilize the advocates, because they have the power, they can march on the Hill, they can mobilize their tens of thousands of members, and they have the power of the checkbook with the research community, and they can say, no, you will not get a grant unless you tell me that your data are being collected in standards-based ways and being facilely shared and distributed.
So the job. I could probably teach a course on that. So what defines a job? In my presentation, when I used the job, I really meant the traditional scripts of HPC or grid sense scripts. But actually, let's say in camera, what comes as a job to the scheduler, in addition to data queries and searches, is a workflow submission. So then in turn, the workflow system will go and communicate with the scheduler and spawn more jobs. That's why when we have 1,500 job submissions, it goes and forks out another million or more, actually. And the definition of job is changing as well. What executes as that little atomic block of execution is not just a script anymore, but it could be a workflow itself. So let's say when we execute actually workflows on Hadoop, what executes as a map or reduce job is a workflow, a sub-workflow, the way we call it. And so what does that buy you in general is you don't have to build scripts for map or Hadoop to wrap, or map or reduce to wrap the code, but you just encapsulate it as a reusable one, so you can use it in different contexts as well. That's why it can scale across different computing paradigms. There was another one.
So that's it. Okay, it's a really good question. Yes, supercomputing, that's why we are getting into virtual machines and cloud-like services within the supercomputers to address smaller communities or non-traditional HPC users. But with the current state of art, how the applications are written, you need supercomputers to solve big data problems, right? That's why we need to invest more in algorithmic development for this next generation for faster analysis. I think that's how I see that.
You were so efficient getting all 4 questions before anybody else has a chance. Why don't you go on up?
This is a workflow question. This has been a good meeting for thinking about workflows. The question is, how do you identify them? How do you link them to the resulting data? It seems like they're very important in the metadata. How do I know where this data came from? How do I know what's been done to it without being able to look at and understand the workflow that it's passed through?
Mm-hmm. So how do you identify a workflow? You know it when you see one. In addition to that, basically generally it's a repeatable process across different executables and data. And so if you're just getting, let's say, data from a sensor or a device instrument and putting the data in a searchable form or in a standardized way into an archive, it is a workflow. It's not, it doesn't have to have all those.
How do I reference it?
How do you reference it? So there's a lot of, yes. So there's a lot of actually projects going on in that space, and one of them was mentioned, the Workflow Forever project that treats workflows as actually research objects. So I often say in a platform, you don't have to just have a workflow, have the data queryable. Workflow is a first-class citizen, and it should be treated like data. And the byproducts of workflows is know-how. Every time you run a workflow, actually you have an opportunity there to extend the knowledge base in that space. And right now, most of the workflow systems are making the effort to collect provenance and standardize that provenance. And so workflows have been workflow community have been really active in creating provenance linkages between these workflow systems and sharing that. And the standards development in this have been activated as well. There's a W3C standard that just came out in the provenance space, which partially also talks about workflows and how you link the products of it. But the workflow itself, there are also community archives for sharing workflows. My experiment project have been actually an initial platform to share workflows. And socialize around workflows, you know, create a social networking platform around workflows. But again, now when you go and look for workflows, there is no single platform that you can see, you know, this is my workflow, and I want to publish this, this is how I share it. That doesn't exist. You know, if you want to link a workflow as a research object to a publication, you have to go through a centralized body. Like in camera, when there's a workflow output publication, we actually use the provenance to create a methods and materials experimentally. You know, this is not methods and materials section of a paper. And then they can reference the camera workflows they executed to generate that. And so there are efforts going towards identifying workflows and actually giving them identification so that they could be addressed later. But I don't think there's like a generalized research going on.
Kevin.
Nice set of presentations. I enjoyed this group as well. And this is kind of a combination of a comment and a question, and maybe a question to Jean and to Delsa. And it's teeing off what Marcia presented on the learning healthcare system. I've been tracking that, and when I talk about it, from my perspective as a denominator person, a population health person, it's a learning population healthcare system. You know, most of the dialogue in that initiative is still about healthcare, but as we know, much of health, at least half, is determined by things outside of the healthcare system. It's a wider variety of things. And so to make things even more difficult for DELSA, more complex, we are entering this world of billions of sensors, various sorts of things, to uncover the exposome that I talked about at the beginning of the day yesterday. And so I think this makes it not infinitely more complex, but much, much more complex. And you know, there aren't very many of me at this meeting, and I think we talked about the cell, we talked about the quantified human, and we talked about the quantified ecosystem. I think it's time to be bringing the quantified ecosystem in sooner rather than later, because these things have influences on everything that's being talked about here today, at least by my understanding of, you know, the biological mechanisms. And it's the only way we're going to tease out these mechanisms of the influences of stress, of violence, of poor air quality, of you know, radiation fields, a variety of things on these deep biological things that everybody here knows so much more about than I do. So I think I just think if in fact DELSA can span from cells to society, from the genome to the exposome, everything, it truly will wrap its mind around this. And actually, I've kind of walked the walk. I've been a long-time NIH researcher. Actually, I started CDC funding a long, long time ago, moved to NIH research. I now got my first NSF grant through the SICE, the Smart Health and Wellbeing, and we're focusing on trying to develop what we're calling a whole health information model. It includes everything that influences health, from all the stuff at the biological level to all the stuff at the macro level, and pull those forms of data in and make sense of it. And to do that requires dealing with lots of different forms of data, but digitizing as much as we possibly can, and dealing with other things. So anyway, it's kind of you know, I'm kind of preaching, but I'd like to see DELSA actively reach out and expand, because this is traditionally the public health community, the environmental sciences community. There are some really bright scientists that are out there working in this area, and they can't accomplish what I think the vision of DELSA is unless they're connected in some way. So I'm looking to you guys to help make that happen.
I would second that thought, and if you have an interest, one of the first projects of our coalition will be to do exactly that around a medical condition, to draw in not just the biological or clinical data, but the public health data and the environmental data and so on.
Okay, my turn. Jeffrey, you talked about dark data, and I'm pretty familiar with that because we have a curriculum clearinghouse that we set up through the National Digital Library, an NSF project, and you know, you could not find any of those things by using Google. So, but none of us have the skills necessarily to perhaps convert our materials to the materials you talked about. Is there some system that you would set up that people could, I mean, some way to you know, convert what exists to what you're proposing?
Yeah, so I mean, what we've tried to do, one of the other things, as you mentioned, again, it's very difficult for a lot of these groups to, move data into a lot of these new formats and, nano publications, semantic web standards and all that. So, what can you do to sort of lower the bar for these people to make the data more discoverable? And so, the platform that we've put together, allows curators from these different communities to come in and help out these data resource providers, in being able to curate the data at a number of levels, and then provides a search interface on top of that so that you can get sort of a community-focused search portal across the different data. And what we're doing now is then turning that data around and to try and expose it better to Google as well. So we just released an antibody registry, sort of an open source collection of more than 2 million antibodies. And we're now sort of the second hit behind a lot of the big core manufacturers. But that also means for a lot of the little labs. So we're getting very high Google hits for, just like Yelp does for restaurants. So for a lot of the big antibody vendors, we come up right behind them on Google. And then the counterpoint to that is that for a lot of the smaller labs that don't have the visibility on Google for these antibodies and things like that, our hits are also very high for these antibodies. So, there are a lot of things that one can do to, both make sort of community-specific tools for search that can be enabled, that we provide. But then there's also this other sort of service in terms of republishing data in new ways, in ways that can leverage Google, leverage some of these semantic web technologies, using some of the the micro-format annotations within web pages to also make these resources more findable through other standard locations as well.
I guess actually what I was wondering is, here's an example. If I were to sign up for LinkedIn and I were to mistakenly click the thing that says, look at my Google email, it would pull in my whole email address box and send out requests to everybody I know. What I'm wondering is there some kind of service, maybe DELSA, this might be a role for DELSA, where a group like ours that has a curriculum clearinghouse already with NSDL could just type in our URL, and your service could convert all of our learning objects to something that we could later go on and curate, but it would be findable.
Right, so that's sort of the first stage in this process where we have an open source catalog of all resources, and then communities can go in and pick out what resources they want to expose at deeper levels. And so, the first place to go is to go to, the catalog and register there. So.
And you'll give us the URL for
yes,
that
yeah,
catalog?
I can. Right now, it's the primary catalog is at Neuroscience Information Framework, but we're rebranding to a generic name since we're also doing diabetes, kidney, aging, animal models. So, and we're in the process of setting up that generic site right now. But.
Great, because I could envision a role for DELSA as kind of a clearinghouse for some of these services. And another question I had is for Lennart. When you're talking about looking at phosphorylation sites on protein structures and things that you might want to correlate, things that you think are there with what exists, have you considered the idea of enlisting students?
Well, this is a very nice question. I like it because one of the most popular crowdsourcing projects, I'll go beyond students, I'll engage everyone, is Folded, which is protein folding, right? And it requires you to actively do something. It's not CT at home where you just have your screensaver do the work for you. No, you actually have to do something. Now, for the phospholocalization, the computer can do it faster than the human. So it's not actually particularly well suited to replace computers with humans. So if you want to get humans involved in doing something like this, you want a task that they can outperform the computer at. But we're trying something else. We're currently trying to build a game on mobile phones and iPads for de novo sequencing of proteins, of peptide sequences. So this big difference between the two curves, right? One of the big problems is that a lot of people are researching very interesting organisms that have not been sequenced. And so we don't have a genome sequence. And we know all these problems. We've heard about the metagenomics as a typical example. But also a lot of higher organisms, complex organisms, parasites, you name it, especially diseases as well that strike people in the third world. They are not sequenced. And so humans are very good at picking up patterns in the spectra. The thing is, you mustn't make it look like a spectrum, because if you show it as a spectrum, everybody will go, oh, this is geek stuff, I don't care. So I asked my people, it was a very silly challenge. I told them, make this into a game, make the de novo sequencing into a game. And they came up with a bunch of proposals. And one of them was you show the spectrum like a building lot, and that has certain allotments available. And then you need to put skyscrapers onto this lot, and the skyscrapers have a certain width. And the intensity of the signal corresponds to how high your skyscraper will be if you place it in a certain location. And that works extremely well. And it's great fun. It has nothing to do with mass spectrometry, but it gives, in the trials that we did so far, it actually gives very good results. And so it's a beautiful example of this, because the human can very quickly come up with an original, interesting, and pretty good solution. And the computer cannot, but it can very easily verify whether the solution is a good one. Right? So the human does something, computer validates it. Now we're going to try and add something new on top of this that I haven't seen, because Folded works with leaderboards, so you try to get the competitive element in there, and then people start logging a lot of hours. What we're going to do is we're going to have the computer help the players. So if a player comes up with a solution, the computer can then use this as a seed solution, and then figure out whether it can do better based on that initial solution. And very often it can improve it a little bit, or sometimes even a lot. And if that happens, the user high score should change, and the user should go higher up in the leaderboards. And so that's another little twist that we try to put on this game, which will be that users actually cooperate with the computer in achieving this goal, and together they can achieve a higher score than they can individually. So you could be third ranked on that spectrum and suddenly go to first rank because the computer realized that your solution was a good one. So that's one way that we're trying to get everybody involved on a specific task, and I hope it's going to be fun and a success, but we'll see what happens.
That's really cool. And if you want students to test it, I can help with that.
Okay, great. Yeah.
I can help with finding them. What's it? Are we done? All right. Jean tells me we're done and it's time for lunch, and I don't want to stand in the way of food.